Today we would be studying cell structure and organization, the chapter 1 of this book, Biology Matters, of course. The chapter 1 is cell structure. Let's write it. Cell structure and organization. Well, shortened form, but yeah, organization. Well, what's in this chapter? So firstly it comes, what are cells? Firstly you have to answer what are cells. So can anyone tell me what are cells? Well, a cell is a unit of life and it consists of living matter called protoplasm. So let me bring you this book. So what does a cell consist of and what is a cell? Well, a cell is a unit of life, consists of a mass of living matter called protoplasm. So that is kind of a bookish definition. Let's go back to what is a cell. So a cell basically means a factory. It means a factory where all the biochemical processes that occurs in your body, they happen to occur inside the cell. And the cell consists of, what does the cell consist of? It has cytoplasm, cell surface membrane, and most of them has nucleus. But of course some of them, they don't have any nucleus, such as the red blood cell. They don't have any nucleus. I would be going into details why. And each living cell consists of a living material that is called a protoplasm. So what is the protoplasm? Protoplasm is the gel-like substance that is present in the cell. Okay, the substance that is gel-like. For example, everything in the world isn't hard. It is either solid, liquid, or in gaseous state. So here protoplasm is a jelly-like substance that makes the cell look like a jelly. It's not hard, it's not very soft, it's just the right. Alright, it's just right, and it's just like cell. And the term cells, and the cell was first discovered by Sir Robert Hooke. He was an English scientist. What he was doing is, like, he took slices of cork. So cork doesn't mean the badminton cork you're playing with, it's a plant thing. So he took thin slices of it. So as you know, we have to take thin slices of anything if you just want to examine it. He took thin slice and he placed it under microscope. Well, of course the guys in 1667, they did not have microscope back then. So the guys in 1667, they did not have microscope back then. So what did they do? They just used whatever microscope they had. They had a simple microscope at that time, definitely not like the very complex microscope that you would be seeing in your school laboratory. Well, so at the time he just casually took up a microscope and thought, how does a cork looks like? Interesting. So he decided to zoom it in. When he zoomed it in, he simply just, what did he see? He saw simple blocks like this. Alright, he just saw some blocks of cells like this, like some blocks like this. He called these blocks cells. cells, cells. So you can see, I guess, this picture right there, tiny blocks. These he named cells. So from that he just got the name, and the cells got the name cells. Alright, and of course, why couldn't people before him? Cannot say. Because probably they had other works in their hand to solve, I guess. And cells are so small that you cannot see it with your naked eye. So for that you have to use microscope. So currently there are two famous microscopes are in use. One is light. Now there is electron. Well, these two names are given, of course, from the medium the media that is used to inspect the things that are put under them. So these things are named after that. For example, in light microscope, light is used to inspect a specimen that is placed under the light microscope's lens on a slide. And of course, in electron microscope, electron beams are used to zoom in and look at the specimen that is entered in the electron microscope. And in light microscope, there is simply one type of light microscope that I would like to show you. This is a light microscope, simple light microscope that is currently used actually in our school as well. It has a simple mirror, it has eyepiece lens, it has some knobs, the fine adjusting knob, the coarse adjusting knob, and the 3 lenses. This is the tray where specimen slides are put, and there is an aperture through which light enters, and it straight goes to our eyes. Alright, this is a light microscope. So it uses light. Light is comes from the sun, gets reflected in this mirror and enters straight through it and reaches our eye. As we know, when light is reflected off a surface, we can only see the object when light is only reflected off the surface. Otherwise, we won't be able to see it, because our retina just makes an image of it, upside down image, that you would be learning later, that it makes an upside down image at the back of our eye, and that is corrected by the brain, of course. And the image signals are transported from our eye to the brain via optic nerve, where the image is corrected the right way up and then visualized. Well, let's go further into the electron microscope. We have talked a lot about light microscope, and of course light microscope can magnify an object up to 1000x, as it's written there, 1000 times. Well, an electron microscope, as you can see, the big boss can magnify it up to 200,000 times. A lot, right? 200,000 times, because it, and of course of these two, the big boss has a greater resolution. Well, for example, you can see cells in light microscope, but you cannot see most of the organelles in light microscope. And what do I mean by organelles? Organelle means simple organs that are found inside the cells are called organelles. So as we speak of organs, say we have liver, we have pancreas as our organs. So like that, there are some organs inside the cell that carry out major functions. So that organs inside cell are called organelles. Well, okay, so let's go to electron microscope. The electron microscope are of two types: TEM, SEM. Only for your information, this TEM is known as transmission electron microscope. Transmission electron microscope. SEM means scanning electron microscope. And with the name you can see, transmission means electrons are transmitted through the specimen. There is a specimen, suppose a specimen, electrons are transmitted through it, reflected off a barrier, received by the receiver, or receiver might be there as well. So my bad, receiver isn't here. Well, electron beam is transmitted, passes through the specimen, and the the receiver, the receiver receives the electron, and if the electron takes more time to pass through an object, that means it is more denser, right? So as we have seen in the, what is that called, the sonar system, the sonar system, that the ultrasound reflecting system that ship uses. Okay, so it passes through it, and the electron count per unit time is seen with this receiver, and we get a clear picture of this thing, like how thick is it, what is the shape. And scanning electron microscope, it simply just scans the surface. All right, so here is a specimen. Say this is not insect, I have to draw an insect properly. Well, I can't draw properly. Well, okay, suppose this is an insect. I don't know what insect that is, probably a ladybug. All right, so basically if you scan this insect, or maybe it's a lice, if you zoom it in with electron microscope, you would only be able to see its outer surface properly and clearly. All right, but you won't be able to see the inside of it. Like for example, in the inside, for it has a heart, you won't be able to see the heart. But in case of scanning electron microscope, you would be able to, in case of transmission electron microscope, you would be able to see the heart. That means electrons passes through the specimen in transmission electron microscope, but in scanning electron microscope, electrons cannot pass through the specimen. So only you get the proper idea of the shape of the specimen. And well, in light microscopes, color images are seen, and for enhancement in visualization, dyes can be added. Well, dyes are chemical substances, so those are used to highlight important parts in light microscope. And in electron microscope, you get a black and white image that can be colored through computers. So of course, in electron microscope, you don't get a simple image, right? You just get a micrograph that is visualized using a computer, and in that computer there are some softwares that can auto color the parts you want to visualize. So the parts are well distinguished, Well, now we have this, like how to cut a cell, for example. Well, let's say I have this human. All right, I have this human, and I cut him like this, straight through the middle. So when I cut him straight through the middle, he would be cut into two parts, right? If I cut him again, two parts. So what will I get here? If I take this thin part, inside part, what will I get? I will get his brain, I will get his probably heart, lungs, kidneys, stomach, and okay, whatever that is. Well, I would get everything right here, right? So this type of things are called, what is it called? One is longitudinal, one is transverse. Tell me which one is it. How is a man cut? Anyone? All right. So in that sense, you have, this is longitudinal section, or LS. This is longitudinal section. And say I have another human. All right. Say I cut him just like this. Ooh, dead. I just cut him in two halves. All right. So in that sense, if I cut him like this, well, okay. If I cut him just like that, I would get his stomach only, right? I would only get his stomach, right? Maybe kidneys as well, but I won't be getting his brain, his heart, his bladder, nothing else like that. All right, not the lungs. I would only be getting his stomach. So in that sense, this type of cutting is called transverse section, or TS. Transverse section. All right. So this is what transverse and longitudinal sections are? Well, this is a cell, a generalized cell. All cells definitely aren't circular. Some are definitely of, rectangle shape or whatever shapes, I do not know. And cells can change their shapes, the red blood cells, they can obviously, they have to squeeze through the capillaries. That means small pipelines through which they have to move. So they have to change their shape definitely, right? Well, this is a cell, and I told you that cell has organs. When a body has some organs to carry out some special functions, or body has some specializations in it to carry out certain functions, that is called organ, that are called organs. And when the cell has some specializations, that are called organelles. Alright, so let's check out some organelles right here: mitochondria, ribosome, etc. Let's see the labeling right there. It often comes during your exams, right? So this is the outer layer, outer boundary, that's called cell membrane. It can be also called cell surface membrane, and this is obviously a permeable membrane. Well, it's a partially permeable membrane. It is permeable, but not to all. What I mean by partially permeable is it lets certain substances in, but not all the substances in. Alright. For example, your guard of your house. Does it let everyone to enter your house? No, right? First, you have to confirm that you know the person and he is safe for the building. He won't cause any damage to the society or your colony. Only then the guard would let him enter, right? So this is the guard. He won't be allowing to enter anyone who wants to enter. He will be allowing only the selected persons. So the cell membrane, cell surface membrane or cell membrane will only allow the persons that are allowed to enter the cell. And well, the allowed persons must be small, they must be maybe they may be water soluble, they may not be water soluble. Well, this cell membrane is made up of a substance that is called phospholipid, as you might see later on. Phospholipids are like this. Alright, point. These are guards, alright? Yeah, these are actual shapes. This is how, wait, I drew the other way around. My bad. The phospholipid bilayer, probably it's like this. Yeah, something like that. Just messed it up. Well, you see, this is the phospholipid bilayer. This is the lipid tails. The hydrophobic they are called, and that is the hydrophilic. Well, these are some stuffs that you will learn later, but the structure looks like this, simple. Some army personnels, they are just standing as guards. Alright, well, let's go for mitochondria. Well, what are mitochondria? Well, at this stage you can definitely say they are powerhouses of the cell. Yeah, they are powerhouses of the cell. So mitochondria, they are simply double layered. Alright, this is a kind of mitochondria that I drew. First layer, then another layer. These are the foldings. They have some names, These are called cristae. This is the outer layer, inner layer, and inside this there are some things called ETCs found there, electron transport chains. So yeah, this is it. And they definitely, the inner part, it has some sort of liquid or jelly like substance called matrix, the mitochondrial matrix. Alright, yeah, that is mitochondria that you need to know right now. It is the powerhouse of cell. Why? Because inside that ATP can be made, or ATP can be broken down. What is the ATP? ATP is like battery, adenosine triphosphate, ATP. It's like a simple battery that is stored in this factory, and when needed, this battery can provide energy when needed, and when excess energy is available, this factory can convert the energy to battery. Alright, the batteries can be made or destroyed at any time when the body needs. Alright, well, now let's come to chromosome. Chromosome is something that holds you from your birth. You have all your information. It's kind of the cell archive. It's a cell archive where all your hereditary informations and how the cell will grow, how the cell will function, everything is stored in the chromosome. Afterward, it comes the nucleus is covered again by another layer called the nuclear membrane, and this tiny thing is nucleolus. Okay, and this is chromosome, this is nuclear membrane, and this thing is called nucleoplasm, the inside matrix thing, the liquid part on which nucleolus and chromosome they're kind of floating. That is called nucleoplasm. There comes cytoplasm, and cytoplasm is the name for the liquid like substance that is found inside the cell, alright, on which the substances are floating sort of. And then comes the nucleus, of course, the central part. Then there are vacuole. These are definitely temporary vacuoles that are used to store some temporary food materials or other materials in the cell. These are ribosomes. Those are needed in protein manufacturing. Well, there are two types of ribosomes 70S and 80S, that you will learn later. There are two types of ribosomes, 70S and 80S ribosomes. Alright, I think that's it. I think that's it. And also some other components can be found inside it, such as this endoplasmic reticulum. There are two types of endoplasmic reticulum, namely rough and smooth. The smooth is called SER, rough is called RER. Well, the smooth endoplasmic reticulum, when the ribosome is attached to the smooth endoplasmic reticulum, what I am trying to say is, where's my pen? Okay, found it. Well, say this is the endoplasmic reticulum, sort of. Say this is the endoplasmic reticulum. When this is like this, it is called smooth endoplasmic reticulum. But when ribosomes are attached to it, right, its surface seems to be rough, right? That is when it is called rough endoplasmic reticulum, or RER. And there is also Golgi body or Golgi apparatus that is used for protein modification. And there are some vesicles that are used to transport the proteins or the substances made inside the cell. It transports it from inside the cell and fuses with the cell surface membrane and excretes it on the outside. Okay, and sends it or releases it to the outside. And I've told you, ribosomes attached to the membrane, and there are some free ribosomes, so that makes up the attached ribosomes makes up rough endoplasmic reticulum. Well, in animal cell we have cell membrane only, but in plant cell, as I have told you, the organelles that have some special jobs to perform. Well, in plant cells there is cell wall, there is cell surface membrane, also there is cell wall. The inside part is the cell surface membrane or cell membrane, the outside part is the cell wall, and the cell wall is made up of a component named cellulose. It is made of a component called cellulose that we animals, or the humans or animals, that cannot make. Alright, so that can be only made by plant cellulose, and that is used to strengthen the plant cell. The plant cell is quite strong, so that's because it has cellulose cell wall. Well, besides, in plant cell there is also a thing called chloroplast, and chloroplast contains chlorophyll, and you all know probably that chlorophyll can trap light and can use the light energy in photolysis. I think you're familiar with the name photolysis, using light to break water, photolysis. Well, that energy, that light energy, is used to make their own food, such as glucose. For an example, that glucose is converted to sucrose and later on to starch that is stored and that we humans eat from plants, and we get the starch and we then reconvert it to our other forms that can be used, say glucose that the brain needs to carry out its function. Well, as I've told you, the cell membrane is made up of phospholipid bilayer. That means lipids, or you can say fats. So it's made up of lipids and of course some proteins that allow substances to pass through it. Definitely it must have a side gate, so the side gates are made by proteins. Well, what I mean is, I've told you these are the guards, right? So there are some channels present inside the guards that are also guarded, but yeah, some channels present through which substances can pass through, right? Well, I think you have already known what the nucleus is, but what does it do is, nucleus controls all the cellular processes inside the cell, and also the nucleus allows the cell to divide, and it's because of the chromosomes the cell divides. What does cell divide means? One cell becomes another, forms a copy of its own. Then the two cells divide and forms two other cells. One cell that extends like this. Say, how does a cell divide? Well, that is the process of cell division that we will be learning later. Well, suppose this is a cell. Okay, I'm just drawing it like that. A cell. It forms another part of it like this. Okay, forms another part of it. This part disappears, of course. Then the nucleus is formed, and it forms, then later on it starts forming some kind of walls in it. When the walls is fully made, the cells pinches off and they form two different cells. So this is how from 1 to 2, 2 to 4, 4 to 8, 8 to 16, they just go on dividing, and you are what you are now. Once you were, once you were a small cell or organism in your zygote, of course, and afterwards you became you. So what you are right now. So definitely that's a result of cell division. Well, the chromosomes. Now we have come to the chromosomes. They contain parts called DNA. The chromosomes are made up of DNA. Okay, so what is a DNA? DNA, also called deoxyribonucleic acid, the full form, DNA. Well, as you have seen, it's like spaghetti noodles, right? It looks like noodles. Yeah, it does look like noodles, but later on when it is in dividing phase, it just forms kind of like earthworms. All right, they just swell up and they are like earthworms. When you try to visualize this under light microscope, they're visible, but yeah, like earthworms. But in chromosomes, that part is called chromatin at the time, that dividing stage. Of course, at that time it is called the dividing stage. It condenses and swells up, it condenses and thickens. All right, that is a word. It condenses and thickens. Well, this is from another book. Well, you might not know it. It's from Olevels. It's I mean Alevels. This is this book, the course book for Alevels. Well, here you can see the double helix structure. This is called the intertwining of these two strands. They just form a double. This is called a double helix structure, and this thing is DNA. And this double helix structure, it's wrapped around each other and wrapped around some proteins as well, and they just simply form the chromosomes. And the proteins they wrap around just are like some potatoes. Those are called, I forgot the name again, I just remembered it right now. Well, whatever, there is some protein and it just wraps around the protein and forms the, and you know, there are several rounds of it and forms the chromatin structure. Oh well, I remember that. It's histone protein. It's wrapped around histone protein and simply forms the, you know, the DNA structure. And the DNA, if you want to know better, it's not in your syllabus, of course. It's like this: sugar phosphate backbone, phosphate, pentose sugar, 5 carbon sugar, and nucleotide base and nitrogenous base is added to it, and this is how DNA forms. And there are 2 strands of it. They intertwine with each other, they wrap around themselves around a histone protein, and they form the DNA like that. And it's inside this DNA, it is coded how you will behave, what the color of your hair will be, color of your iris would be. All of these are predetermined in your DNA. That often has hereditary informations in them that comes directly from your mother and father. Well, now let's look at rough endoplasmic reticulum. It has ribosomes attached to it, and ribosomes, definitely they are the sites for protein synthesis. They are where proteins are made. And in smooth endoplasmic reticulum, that you can see right here, ACR, in smooth endoplasmic reticulum, lipids and steroids are made, such as, you know, cholesterol, and some reproductive hormones such as estrogen, testosterone, etc. And it is a major site for, you know, calcium ion storage. So calcium ion is needed for muscle contraction, that you would know in your later life if you carry on with Cambridge. Well, yeah, it's for calcium storage, and from this, you know, it stores calcium in it, and that calcium can be used for muscle contraction. That means the more calcium you have, the more stronger you are. And the medicines you take, you know, the drugs, the medicines you take, ACR is responsible for the drug metabolism. ACR is needed for metabolism, and as you can see, yeah, it converts harmful substances into harmless substances by a process called detoxification. That is also done in the liver. Liver detoxifies, you know, your body. It detoxifies your blood. So yeah, ACR here, ACR does that. All right. And here is Golgi apparatus. Well, what does Golgi apparatus do? Simply just modifies protein. So it's kindly not in your syllabus, but let me tell you, proteins are like some threads like this. All right, single threads. So this long thread, okay, this cannot be used by the body, or this is too big for the cell. All right, so it cannot just let it like this. So what the cell does is, like, so for example, you have a long rope lying around your floor. So what would you do? You will just simply coil it up, right? So the cell does the same. It just simply folds it in. It doesn't have a brain, right? So it cannot fold it properly. I mean, coil it. So it just folds it in different structure, folds it in a compact shape. This folding is done in Golgi apparatus. And after this folding is done, say this is the protein. So when you go to a market or a shop, so whatever you buy, they just wrap it in a poly bag, right? A bag, a covering. So yeah, So from Golgi apparatus, a covering just is given to protect the protein or say some other substances that travels inside the cell. So this protective layer is simply this total thing is called vesicle. So what does it do is like it simply moves on to your guards. All right, simply moves on to your guards that were stationed, and with that guard it makes a deal, and then this vesicle simply opens up like this, and the protein in it simply moves out. This is called pinching or exocytosis. It just pinches off from the Golgi apparatus. It is released from the Golgi apparatus in a vesicle, and it fuses with the cell surface membrane, and this thing just leaves the cell, and the process is called exocytosis. It requires energy definitely, so ATP is needed. So ATP is found in mitochondria. Mitochondria makes it or stores it. So yeah, mitochondria is in use. They are sending ATP and saying, guards, please use this energy, yeah, to move apart, and let this vesicle empty itself and let the protein get out by exocytosis, right? So that is the thing that occurs at there. Here you can see the some forms that is given here. Vesicles pinching off and then fusing with Golgi apparatus. From Golgi apparatus, vesicles being pinched off, and the secretory vesicles fuse with the cell membrane and releases the substances by exocytosis. Well, then comes mitochondria. It is where aerobic respiration occurs. What does aerobic mean? Aerobic means air, and what does air means here? It requires oxygen. Why is oxygen needed? Oh, we know humans need oxygen to live, right? So why do we need it? Well, it's not in the syllabus, but we need oxygen to, well, I'm sorry for distraction. Well, yeah, so back. Mitochondria right here, it works in aerobic conditions. Well, what does aerobic condition means? It needs oxygen. So have you ever wondered why we need oxygen? We all know that we need oxygen that we get from plants, but have you ever wondered why oxygen is needed? Well, where is the mitochondria that I drew? Well, my whole sheet is a mess. Let me find my mitochondria first. Where is my oh wait, there is my mitochondria. Well, remember I have mentioned about something called ETC, electron transport chain. In this electron transport chain, a process called oxidative phosphorylation occurs, in which oxygen is needed. So that is why we need oxygen. Okay, if oxygen wasn't present, so ATP would not be made. So ATP wouldn't be made right then, and we won't be getting that much energy, and we won't be able to carry out our metabolic functions. So we would simply die sometime later, not instantly, of course. And now from animal structure, let's just go on to plant structure, the plant cell structures. As you have seen, it is trying to show some differences between animal cell and plant cell. Well, in plant cell, I have already told you it has chloroplast and it has a cell wall extra. Besides, in plant cell there is a large central vacuole. This vacuole is there to press the components right against the cell membrane. And you know why? That is because light definitely enters the cell through these cell walls, through these cell membranes. Alright, if the chloroplasts aren't present nearest to the cell membrane, so they won't be receiving as much light, So they are kind of pressed hard against the cell surface membrane so that they can easily absorb the lights that are coming from the sun. So the chloroplasts can use the energy in reaction centers, and they can just make ATP from there for photosynthesis. Well, photo means light, synthesis means making. So using light to make substance. And here we are reading further what is chloroplast. The oval structures found in cells. It is green, and there are some other pigments in the chloroplast as well. There are several types of chlorophylls such as chlorophyll A, B, xanthophyll, beta carotene, etc. Those you will learn later, of course. And yeah, that's it. That's chloroplast structure. And well, let me draw you another structure of chloroplast that is more simplified. Where will I draw? Okay, wait. This is a chloroplast. Inside it there are some balls like that. All right. So, bubbles, interconnections, and inside them, you know what's inside? There are stacks, thylakoid stacks. These are thylakoid stacks. This is grana, this is stroma. Oh wait, I just messed it up, so let me tell you again. Well, I found it easier to visualize. My drawings probably aren't that visible that much, so yeah, that's it. This whole stack, the whole balls that I drew, these are grana, and these stacks are thylakoid stacks, and this matrix is stroma. And definitely chlorophylls are found in the thylakoid stacks. You don't need to worry about that, you would be studying it later. This is the structure of a chloroplast. Alright, the chloroplast that you are seeing here, this is the labeling structure. This is from the book, the Olevels book. So you don't need to worry about that. I just showed you this to visualize it. And yeah, now we have come to vacuoles. Well, it's a fluid filled space enclosed by a partially permeable membrane. So it simply has a partially permeable membrane through which everything cannot enter, but some of the things can enter that are only allowed by the cell to enter. Okay, that is very selective. It's a selective barrier. And the plant has a large central vacuole that contains a liquid called cell sap. In that cell sap, it maintains the turgor pressure, of course, so that the chloroplasts are pressed against the cell walls, and so that they can get enough sunlight, so they can photosynthesize properly. Well, here are some differences between plants and animal cells that you might be asked to write about. Well, in plant cell wall present, and in contrast, animal cell does not have cell wall. Plant cell has large central vacuole, but animal cell has small temporary vacuoles. And in plant cell, chloroplasts present, but in animal cell, chloroplasts aren't present. So these are some differences in them. Well, I have talked at the start of the class that there are some things that I will mention, that how cells, they just pass through tight pipelines called capillaries, right? So for here, that the process when a cell becomes specialized for a special function is called differentiation. Yeah, the differentiation, there is a differentiation in maths, in calculus, differentiation and integration that you will learn in pure mathematics if you carry on with Cambridge. And we can see there's lots of cells in the body, and they have many different functions right there. So some are specifically adapted to carry out their functions. For example, the red blood cell. I've talked about red blood cell not having the nucleus. You know why? The nucleus isn't present here to allow more hemoglobin, to let it contain more hemoglobin. So the nucleus is removed from its place. Why? So that it can contain more hemoglobin, so it can transport more oxygen. and besides that, it has a biconcave shape that increases its surface area to volume ratio. So what does biconcave mean? Biconcave simply means this part is thicker, this part is well, let me use my marker, this part is thicker than the inner part. Alright, that part is thicker, the outer part is thicker, inner part is thin, the outer part is thicker again. This is called a biconcave shape. Alright, and what does that do? That increases the surface area to volume ratio. And what is the surface area to volume ratio? I think you have studied already surface area in maths. Well, surface area simply means the area of the surface. For example, this let me draw a cuboid here. Not a cuboid, let me draw, wait, is it alright? Yeah, it's a cuboid then. Well, in this cuboid, the total amount of air, the amount of air that the box contains, or amount of water, or amount of anything that the box can maximum contain, that is called its volume. But suppose the box is made from cardboard. Alright, box is made from cardboard. Okay, cardboard. So if you want to make a box, you have to go to a shop and tell the shop owner that I need some amount of cardboard, right? The amount of cardboard that is needed to make this box is the surface area, and the amount of air or water this cardboard box, the box made from cardboard, that it can hold, that is called the volume. So the surface area simply means the area of the cardboard, this cardboard, not the inside air, the cardboard only, the cardboard, the area of the cardboard. Alright, so it simply means to increase the surface area. And how the surface area is increased, you see, in case of this box, okay, that it simply requires, say, 5 to 10 centimeters square. Its surface area, let's see, okay, let me make a cuboid, then you would be it would be easier to understand. My bad, sorry, I'm really sorry for that. Well, yeah, this cardboard is of 10 centimeters square. Alright, this is of 10 centimeters square, and this is of 10 centimeters square. But if you try to make the same cardboard box a bit wavy like this, you would see that this cardboard, okay, these are of the kind of, okay, same, apparently same. Alright, these two are apparently same, but if you're gonna measure the surface area properly, okay, using other instruments, you will find that this thing is greater than 10 centimeter. It is greater than 10 centimeters square. Definitely it is greater, but I told you they were apparently similar, but due to the presence of this wavy structure, surface area increases, but the volume remains same, right? This cardboard box will contain the same amount of air as this cardboard box, but it requires more cardboard to make this box. So that is why it has more surface area. This increases the surface area. So more surface area means more hemoglobin can cling to the surface, more oxygen can be carried. Alright, and oxygen can diffuse in it quickly, very quickly. So inside part is the thinnest part, so oxygen can diffuse in it more easily, right? It can get into it more easily. And it lacks a nucleus, I have told. It's flexible, and the flexible part I have told you, say this is a red blood cell. Alright, and I have this pipe. How can the red blood cell just get into this pipe? No idea, right? If it were a ball, it would get stuck. It would just, it wouldn't just get into the pipe. But in case of red blood cell, it can change its shape. It can just squeeze its way through like this. So this ability to squeeze is called its flexibility. So yeah, it is flexible, so it can easily squeeze through the capillaries. Well, I would be going into the details of capillaries, why the red blood cell has to squeeze through it. When something has to squeeze through a place, its speed decreases, so it increases the ability of it to to exchange some substances that I will be talking in the further chapters, not today, of course. Here we are having the muscle cells. So the muscle cells, they're cylindrical, long cylindrical structure, the fibrous muscle tissue, long cylindrical structures. You see the protein fibers, and it contains many mitochondria. You know why muscles need mitochondria? To contract and relax, so that you can just lift off a bed of your own and just tell your mom, see, I'm so strong, and you can just flex it in front of your friends, of course. So yeah, muscle cell contains mitochondria to provide the necessary energy. The more you exercise the muscles, the more mitochondria it contains, the more energy it can provide, the more the more strong it becomes. And another cell is the root hair cell. The root hair cell is basically found in plant cells, in the root of plants. So it is long, narrow root hair. So this cell simply elongated itself. It simply elongated its body and formed this root hair cell. The vacuole is elongated. So why is that? To increase the surface area to volume ratio. Okay. So what is that? When suppose this was missing, right? So as you can see, there are only some spaces for the water or mineral ions to enter, but when this elongates, there are more area for this water and mineral ions to get into. So this is how surface area to volume ratio increases the water absorption and mineral ion absorption rate. Well, that's the end of this chapter, and we have come to the end of this chapter. This is the summary of it. As you can see that we have talked about, oh, I have missed one thing. The cellulose, the cell wall is fully permeable, but I have mentioned that a cell contains of two parts, right? I mean, a plant cell consists of two guards. The outer guard is for strength, the inner guard is for selection. The cell surface membrane, I told you it was partially permeable. This cell surface membrane, the inner one is partially permeable. The outer one allows anyone to enter. Okay, this outer wall only has the duty to provide strength, not selectivity. But the inner membrane, provides the selectivity. And yeah, I've covered everything right there. You can just have your revision from here. Yeah, that's the end of this chapter, I guess. Yeah, it's the end of this chapter. So yeah, that is the structure, and I would like to show you ribosome. Practically, ribosome is of this structure. You might see electron microscope. See, this is the electron micrograph of endoplasmic reticulum. Well, you see, this is a transmission electron microscope. It requires definitely a skilled personnel, and this is how the structure is. You will later know of this when you grow up, of course, so it's not for you right now. You see, this is how the light microscope simply works. Light bulb or a mirror simply directly enters our eye, and this is how we just are able to see the specimen, and the specimen must be transparent. I have made several mistakes in the lab. Definitely, I thought at first when I was a kid that any specimen can be visualized under my light microscope, but it has to be transparent. So if light cannot pass through it, you can only study its surfaces, not the insides. If you want to see inside of a cell, you just have to have a specimen that is transparent. So yeah, that's it. Thank you, students. That's it for today. We will meet later with the next chapter. This is biology, of course, I forgot to mention. Well, yeah, thank you. Goodbye. Hopefully you would be coming back to this channel and be with us acing the Cambridge O-levels and A-levels. Thank you, and bye bye. Take care. And please remember to like, comment and share, of course, to your friends so that they can also learn from this. And if you have any other queries, you can directly ask in the comment section, and I will be replying them ASAP. Thank you so much.